Hi everyone, I'm Jules Mary on behalf of GenScript, and today we are interviewing iGEM, University of Paris. So iGEM University of Paris, can you please tell us more about yourselves and your project?
Hi, Jasmine. Yes, very lovely to talk to you today. And well, here there are two of us representing the project today. It's me, Artem, and my colleague Mark. And I think we can share probably some slides to just walk you through what are we trying to work on. Okay, this works. So once again, we are the iGEM team from the University of Paris, and we are kind of spurging. We are started from the BME Biomedical Engineering Master's Program, which is really a joint program between three universities. So it's Université de Paris, it's also Artemisia, which is the engineering school of Paris, and the PSL Research University. And our team was formed primarily with most of the core team members coming from this program. And our project is called ExaSwitch. And what we're trying to address as the problem is the problem of cancer. So one of the main questions when we're talking about cancer is that it's not really diagnosed correctly on time. Cancer remains the second leading cause of death worldwide, despite continuously advanced treatments. There are some therapies which can be effective up to 90% or more in patients that are in stage 1 and stage 2 cancer. However, late stage presentation and lack of symptoms of the initial disease mean that a lot of cancers are diagnosed quite lately, especially in low and middle income countries where the access to different diagnosis tools can be limited. And when cancer is identified early enough, it can respond to treatment rather well. And we can see that, for example, breast cancer or prostate cancer, it can be surgically removed, and then the patients have really good 5-year and 10-year survival. However, the problem is that when the cancer is detected much later, well, unfortunately, the therapy is not effective as much anymore. So when we look into the ways we're detecting cancer, we have our gold standard tool, which is the conventional biopsy. However, conventional biopsy by itself is limited. It's a tool that requires a suspicion of a cancer, because you need to take a piece of a tissue and look at it under the microscope to realize whether the tissue is cancerous or not. So it's really a confirmation tool rather than a screening tool. And it's also an invasive procedure, which is rather unpleasant for the patient because you need to access the tissue, and generally is not possible for all cancers. So in the contrast to conventional biopsy, the new approaches try to look into different samples to diagnose cancer. For instance, patient biofluids, which include blood samples, urine, or even saliva swabs. And in this case, if we use this kind of laboratory analysis, we can perform screening because you can screen patients' blood without suspicion of a cancer. You don't need to access tissue. And in blood of patients, you can find circulating biomarkers of different tumors. And so it's true for other biofluids, depending on the tumor type. And as well, these kind of procedures are minimally invasive. Of course, with saliva and with urine, it's really easy to collect the sample. But even with blood, compared to biopsy, it's a relatively simple procedure that is done for a lot of medical tests already. So when we look into biofluid screening, or liquid biopsies as they're called, we can look into the patient's biofluid samples, and we can see a lot of things going on there. And when we talk about cancer patients, we can also find a lot of different cancer markers in the biofluid samples, starting from just the proteins, hormones secreted by the tumor to interact with the body of the patient, up to the circulating tumor cells at the later stages of cancer. So as tumors shed different molecules into the bloodstream to influence health tissues and to interact with the immune system, they also secrete exosomes. Exosomes are specific extracellular vehicles which are used for between tissue communication in healthy cells as well. And as many other things, they're deregulated in cancer, and cancers use exosomes, as it was shown, to prepare metastatic niches and generally interact with the body. And within those exosomes, you find, again, proteins, DNA, RNA fragments, which can target tissues. So from these exosomal contents, we selected non-coding RNAs that we think are a really good marker, because when we talk about proteins and DNA, those are relatively stable markers, whilst RNA is a more of a dynamic marker that closely reflects the state of the tissue of the tissue of origin, and also they appear to be deregulated at an early stage in cancers. So they are perfect for early cancer detection. Now that we have our targets, we need to move on into detecting them. And our idea was to utilize the whole switch to quantitatively screen for these target microRNAs. Now, the whole switch is something that was inspired by naturally occurring riboswitches, which are found in different organisms. And they were originally used to screen for RNA viruses. So there are antiviral diagnostic tests which already utilize this technology. And the idea is that the whole switch is really just a sequence of RNA, which is bound in this loophole structure. And it has 4 key elements, really. One of them is the whole sequence itself, which is partially complementary to the target non-coding RNA, or just the target. And the other part is complementary to the beginning of the reporter gene, which allows this kind of loophole structure. And as the loop is forming, it blocks the access to ribosome binding site. So the ribosome cannot bind when this structure is compiled together, and translation cannot happen. When the target RNA appears, it binds to the whole sequence of the whole switch, and it induces opening up of the structure. And once the structure is opened up, the ribosome binding site is freed, the ribosome can bind, and then there is the reporter gene translation, which can be detected later on. Now, the whole switch by itself is an interesting technology, but being biomedical engineers, we decided to complicate things a little. And what we decided to do as our project is to manufacture a microfluidic device in which we would put a single drop of our blood sample, and in different chambers we would have whole switches that would interact with specific microRNA targets. And this way we would have one chip that would test for multiple biomarkers of different cancers. And from that, we would be able to potentially detect or kind of highlight that a certain patient can have risk of certain cancers and direct them to appropriate treatment. And so what are we doing now is what we've been doing for a while. Obviously, the first step is to generate the whole sequence because it's not an obvious process. So we computed them and we built our models. So we have our model whole switches. And now we're working on cloning and obtaining the plasmids that will be then producing the whole switches within our bacteria. And once we have finally gotten to our stage of having working plasmids, we will have to validate them against our microRNAs, which we are also separately producing in cancer cell cultures. So that is also a separate part of our wet lab. And this is the timeline, although I do suspect that most of the iGEM teams have very similar timelines. And so right now we are in kind of doing our additional experiments, trying to figure out how to make our toeholds work in the end. And this is the members of our core team who have started and will continue the project now. We also have many other members that didn't make it onto this presentation that take very specific roles in the project at specific periods of time. And of course, we will later on update our roster with everything. And I think this is our last slide with 2 references, which could be interesting to read up to learn a bit more about RNA biomarkers, the whole switch as a starting point. And then, yes, our mail address. You can find us on social media. You can ask your questions.
Thank you so much for sharing your very elaborate presentation on your team's project. It's definitely it's definitely going to change the way that diagnosis is done. Definitely facilitate how people are diagnosed and how soon they can be diagnosed, and hopefully further treatment. So, can you tell us more about your proof of concept process and what it was like?
Well, I'm not sure we can say that it was like because we're still going through it.
Okay.
And of course, as we're going through proof of concept, we encountered problems at every step. Because initially, what we had to do, as I mentioned, is to actually design the RNA sequence, which will allow us to detect the target. And even at that step, we already had a lot of questions of how to, because other teams before and generally other scientists have created tools that can help you design the whole switches to detect different things. But there was only one paper in so far that we found, at least, that used Toll switches to detect microRNAs, and they did it inside eukaryotic cells, well, cancer cells essentially. And so we had to adapt scripts in order to recalculate the model a little bit and to make it work properly. And so we also had our first model that we designed, and then we started looking into it. And somewhere in the process of ordering the plasmid, we realized that mathematically it makes sense. But if you can try to imagine the process, you realize it will actually not work the way we want at all. So there was obviously a piece of code missing from our model. So we had to fix that and that and that. And now I think we have several candidate molecules that we're waiting the plasmids of. And so that's definitely an improvement. And in the meantime, of course, we were also working on actually trying to get our molecular biology to work. And there is obviously a question there as well, because right now we're also working on different PCR cloning and trying to ligate the plasmids to make sure that what we want actually stays together and is usable. And it's not quite working perfectly yet, but we're on the way. And in parallel, of course, we also have somebody who works on the cell culture, because we do need to separate our exosomes. So we have actually our mRNAs that we want to test. And we have separated some exosomes, but then the cell culture got contaminated, then we got another cell culture. It's still a very chaotic process. We see that we achieve certain milestones, if you want. So we get certain results, but it's really hard to predict on how will it go next and at what point we'll actually finalize our proof of concept. Because as you saw in our timeline, our optimistic projection would be finishing proof of concept now, and now we're maybe 1/3 through, maybe 1/2 through. So it's never as quick as you want it to be.
And can you tell us more about the computer model that you use to generate the toehold switch?
So the computer model wasn't my personal part. It was done by a person who's now taking their vacation. But the general idea is that there is a NewPack package, which works nicely with Python, can be integrated. And using this NewPack package, there was previously another team in 2019 that has done a similar toehold, not for microRNA, but for RNA. And what it does is that it essentially takes the transcripts of the target gene, picks up the sequence, does the partial complementary toehold sequence, and from there, it calculates the opposing side, and it calculates the energies that you have when the toehold is kind of held together, and how the energy changes when there is a transcript attached to it. And then there is also a part at which you look at the probability of this binding as well, because of different models, depending on the length of a toehold sequence that is free, and depending on the binding part and the number of GC links in the binding site. So then you get different strength of the connection, and there is a different probability of it opening up. So we even got some models where you add our target microRNA, it connects to the whole sequence, but nothing opens up. So, or sometimes we also got one where we had the toehold that say in toehold conformation, and the probability of that was 10%. So 90% of our toehold would have worked even without the target transcript. So there is this kind of little calculations and things that it checks all the time.
And is there anything your team is doing that's unique for your human practices?
So for our human practices, what we were trying to do, mainly, what we wanted to do is to adapt the design of our microfluidic chip to better meet the needs of patients and doctors. Because one of the questions that we asked to ourselves that we couldn't obviously answer is that whether the person getting tested would prefer to have this test done at the doctor's office. Would it be reasonable to implement it into some routine laboratory checks that somebody gets tested, that their blood for hormones or for whatever really, and then you have this little chip on the side and you just do it as a side laboratory test, or should it be on demand? Or maybe, since it's microfluidic technology and it's supposed to be easily implementable on a single chip used without anything, maybe we could even try to do something a bit like a pregnancy test, that you go to a pharmacy, you can get one, you can put your blood in, you can get a test. And then once we see whether the patients or the doctors see it better as being tested at home or in a facility or with a doctor, then we also want to see how do they want to learn the results. Because again, it's our test will not be definitive, because this will be more of a wake-up call that you should go to this particular doctor, check this particular organ, check this particular set of symptoms. So whether the patients are ready to learn it by themselves, whether there should be some pre-test or post-test consultations explaining them what is going on and what do the results actually mean, and whether it would be better that patients are introduced to the results by a doctor they trust or by some other medical professional. Because what we thought about in comparison was the same HIV rapid tests, because they can be implemented in different ways, but it's mandatory in many countries to have pre-test, post-test consultations whenever you undergo the test, regardless of the result. So what we're trying to do now is we are in contact with La Ligue contre le Cancer, which is the essentially largest charity/research organization supporting cancer research in France. And we interact with them, try to find out whether we could get a chance to talk to doctors and maybe send a survey to patients. So we're now kind of trying to perfect what we're wanting to send out, and most likely we will get to talk to some of the doctors that work with La Ligue to understand what is really the best niche, the best way to implement our project. And so once we have learned that out, hopefully this kind of result of human practices will influence our final design.
Well, iGEM University of Paris, thank you so much for meeting with me today to discuss more about your project and the impact that your toehold switch rapid test kit could have in helping diagnoses be more easily available to the public and hopefully save lives. So thank you so much for meeting with me today and describing more of your project. We wish you the best in the iGEM 2021 Giant Jamboree and in the GenScripts iGEM 2021 sponsorship competition.
Thanks a lot for your attention and interest. It is always really pleasant when somebody wants to learn more about stuff you do.